Hello to everybody. I'm going to present you this artificial and natural cycle. I hope I will give you useful information for everybody to use to their own health. Now, before starting artificial and natural cycle, I think we should understand the physiology of the reproduction. So I'm going to go a little bit in details. So you know that we all have 28 days of menstrual cycles. So when we have an embryo in IVF, we cannot transfer the embryo at any time. That's not the embryo, not on a specific day. You must know that it's not going to implant. So it's either 1, maybe 2 days, but it's a very specific day. So that's why when we do transfers, either frozen or fresh, they are all synchronized. They all have to be synchronized, the embryo age and the endometrium as well. Now, embryo freezing is not a new thing. We do a lot. We have been doing embryo freezing like almost 40 years successfully, and almost now half of our live births are coming from frozen embryo transfers in the world. And we used to have different techniques like slow freezing, but now we have vitrification, which is very successful. We have close to 100% of survival rate to our embryos. When we were doing slow freezing, it was like we were losing one third of our embryos, but with vitrification, it's almost 100%. Why do we do embryo freezing? We have two purposes. One is when we have spare embryos, which is very commonly used, and the other one is which is becoming more and more popular, freeze-all, which I'm going to go into details. Now, when it comes to physiology of reproduction, we have 3 compartments. So the first one is pituitary gland. So now this gland is inside the brain, and it's a small endocrine gland, and it secretes 2 hormones mainly. One is FSH and the other one is LH, as you can see from the screen. The blue one is LH and the red one is FSH. Now, these FSH and LH are affecting the second compartment, which is going to be the ovary. Now, inside ovaries, we have follicles. And with the FSH hormone, what the red one, what it does is it affects the mature follicle, which inside the follicle we have the egg, and it affects mature follicle, and one of the follicles among maybe 20, maybe 30, just one of them develops and it grows. And once it becomes like from 5 to 20 millimeters, then we see ovulation. And this ovulation is controlled by LH hormone. You can see that the LH hormone is like as a peak just at mid-cycle. And this LH hormone just doing ovulation, that means the egg is released from the follicle wall and it enters to the fallopian tube. Now, and after the egg release and ovulation, we see development of a corpus luteum. Now, corpus luteum is the remnant of this follicle. When the follicle ruptures, the egg is released, and the remnants of the follicles becomes corpus luteum. It's a very, very distinguished organ. It's kind of endocrine organ, and it has a lifespan of just 14 days. And if the patient doesn't get pregnant, after 14 days, it just regresses and it just disappears. So it's an interesting thing to see. Now, it's also called yellow substance. Now, these hormones, these follicles secretes hormones. The mature follicle secretes one hormone, and corpus luteum secretes another hormone, which is estrogen and progesterone. And in our second compartment, you can see that estrogen is going to rise until ovulation, and there's no progesterone. It's just at the baseline. But once there is ovulation, you see progesterone. You can see there is a peak after the ovulation. So there is a hormonal effect, which these hormones affects the third compartment, which is going to be endometrium. Endometrium is the lining inside the uterine cavity, and this is where the embryo is going to implant. So there is another synchrony there. So when we have estrogen from the ovary and from the growing follicle, you can see that at the same time we have proliferative phase here. Now, proliferative phase is under of estrogen, under the influence of estrogen. And then once we have corpus luteum, which secreted by the progesterone rise, and you can see that it's called luteal phase, which another name is secretory phase, and the embryo is implanted here. So this is the basic of three compartments. They have to be working all together. So if you don't have this compartment, you will not end up with any of those downstream events. Now, for an implantation, when we transfer the embryo, for implantation, we have a certain name called windows of implantation, the window of implantation. This is a very unique time, and it is just 1 day, maybe maximum 2 days. But if you pass these windows of implantation, the embryo will not implant. So it is very crucial to detect this day. Now, if you transfer a blastocyst a day before, it will not implant. And if you transfer a day later windows of implantation, it will not implant. But if you transfer it just at the right time, then we will have a healthy baby. So it's like in the story of Goldilocks and the Three Bears, you have to be at the right time, at the right place. So this is the basics of physiology. Now, when it comes to IVF, now in IVF, we have, again, three compartments. One is ovarian stimulation, because when we don't stimulate the patient, then we will not end up with multiple eggs. We will just have one egg maximum. And that means that the success and the performance will be too low. That's why we are giving FSH hormone as the physiology. And by giving FSH hormones, we increase the number of the eggs. And then we do egg collection. And after egg collection, we do fertilization by ICSI or IVF, and then we do embryo culture, and then we do embryo transfer. Now, there is something wrong here, because we do the same thing at the same time. We do two things at the same time. One is ovarian stimulation, and the other one is embryo transfer. Now, it's like driving the car and having a conversation on phone. It might be, you might have accident if you do both of them together. So it's the same, because if you have ovarian stimulation, this is the difference of the ovarian stimulation and natural cycle. In natural cycle, here you can see that the orange one is the growing follicle. In natural cycle, you have again many follicles, but just one of them become dominant, and one of them become mature follicle and ovulation. And under this influence, this is the endometrium. The endometrial lining is under this influence, and this is the natural cycle. But when it comes to ovarian stimulation for IVF, one is not enough. So you have to give FSH, and then when you give FSH, many follicles will develop, multiple follicles will develop, and when you have multiple follicles, of course, the hormonal balance, estrogen and progesterone is going to be different. So because like when you measure the estrogen levels in natural cycle, it's like 200 picograms. But when you measure the estrogen cycles in IVF with ovarian stimulation, you have like 3,000 picograms. So it's completely different scenario. So when it comes to like analogy of soil and seed, when endometrium is the soil, it's not going to be the same. So that's why when we transfer the IVF fresh embryo transfer, we have a different endometrium. That's why the performance and the outcome may not be good in fresh transfer. That's the basics of freeze-all IVF. Now, what we do when we do freeze-all, we have segmentation of IVF. So we do ovarian stimulation, we have embryo, and then we do the transfer later on. We defer the transfer which is called freeze-all. Now, currently, not everybody is doing freeze-all, but we have certain indications universally that we are doing freeze-all. One is pre-implantation genetic testing. The second one is OHHS, when a patient has many follicles, many eggs, and she's under the risk of hyperstimulation. And when the patient has progesterone elevation during stimulation, it may affect her success. And when the patient has like structural abnormalities, which is detected during the ovarian stimulation, might be polyp, might be hydrosalpingosis. And when also we do fertility preservation, like social freezing or like cancer patient, we all do freeze-all. But I'm sure in the future these indications will be widened, and probably we will include just for the success rate of universal freezing for all the patients. Now, is embryo freezing successful? This is the national data of US in 2019. According to national data, this is the age group from 35 to 32. And you see on the red line, this is the frozen embryo transfer, and this is the blue line is the fresh transfer, and this is the live birth rate. So when you look at the live birth rate, always frozen embryo transfers are more successful than fresh transfers. And when you look at the age groups, the older the patient group, age group, the success is even higher in frozen embryo transfer. So in some way, this is something successful. And but how common is it to freeze the embryo? Now, we used to freeze the embryos less than now. This is again national data of US from 2005 to 2019. And you can see that in 2005, frozen embryo. Now, in 2019, it's 74%. It's like tripled, maybe more than triple time. So it's becoming more and more common. The reason is why, because the clinicians and the patients are preferring it. It's because it's more successful than before. Now, we are based in Antalya in Turkey, and we do 2,000 a year. And in our center, we do freeze-all. It has been like since 2015. It has been more than 10,000 cycles since we are doing freeze-all. There are almost no exceptions. All the patient goes to freeze-all. The only exception is when the patient wants fresh transfer. So it's very rare. So how did we decide to go freeze-all? Because we analyzed our own data. This is our data. And we published in one of our top journals in 2015. And the red bars are the frozen embryo transfer, and the blue ones are the fresh transfer. Now, these are all double embryo transfer, and these are the combination of embryo transfer for fresh and frozen. Like in one group, 3 BB, and the second embryo was again 3 BB. And this one is like 4 AA, 3 BB, this 5 AA, 2 BB. And you see that in combination of these scores, you can see that frozen embryo transfers were around like 20%, close to 20% higher. That's how we decide. Now, of course, this is our experience, but when we had to go to evidence-based medicine, and the best evidence comes from the clinical trials, randomized controlled clinical trials. Now, in the literature, I'm going to go shortly. In the literature, there are 12 randomized controlled trials, and this is a summary of this graph. But the pink ones, there are pink ones and there is yellow ones. The pink ones, the trials for non-blastocyst stage trial. But yellow all blastocyst transfer. I chose the stage because when it comes to stage of those transfer, the success is different. It's now proven almost that in cleavage stage is equal. So there's no need to freeze the embryo when you do the fresh transfer. But when it comes to blastocyst freezing, the yellow ones, you can see that there are 3 studies. But unfortunately, the 3 studies are not, out of 5, 3 are not good quality. So I'm going to exclude those 2. And these 2, V and Stromland study, these are multi-center large studies. One is from China, one is from Europe. And these are all published in very highly prestigious journals, and they are also in very high numbers. So when I combine these 2 studies, it comes to 2,000 cycles, which are randomized, and you can see that this is fresh transfer is 40%, and freeze-all is 50% live birth rate for single embryo transfer. So the relative means that it's like 20% higher rate in freeze-all. So according to literature, if you are going to blastocyst transfer, it seems like freezing the embryo provides provides better results with freezing the embryo and transferring later, or deferring the embryo transfer. Now, when we have a frozen embryo, we have 2 ways to transfer, because as I told you, we cannot transfer the embryo at any time. We have to prepare the endometrium because it's not under influence of any medication. Now, we have 2 ways. One is artificial cycle, and the other one is natural cycle. Now, as I told you before, this you must remember this graph. I have showed you all these compartments and endometrium, all these together, and in natural cycle, to transfer the embryo according to the ovulation time, because since it is under influence of ovulation, in natural cycle we follow the ovulation, and once we saw the ovulation, which we detect through ultrasound and also blood test, after 6th day we transfer the embryo. So this is natural cycle. But when it comes to artificial cycle, we shut down the ovarian hormones. So we don't want them to interfere our preparation. So we give estrogen and progesterone that are important for endometrial lining. We give them artificially, we give them as medication. So what we do is, this is the critical part. We transfer the embryo on 6th day after progesterone. So once we gave progesterone to the patient, after 6th day, we transfer the embryo. This is the important for dating of the endometrium. It's important for the success rate because we have only one specific date. Now, which one is better? Now, if it comes to practice, I think now 90% of current practice in the world is using artificial cycle, because the reason is it has ease of scheduling, because the patient is happy, she comes, she only has single visit, and also 1 month before, we know that the patient is going to have transfer on this specific date, so the patient can make any plans. So this is something good. But natural cycle, it's not good, because you have to follow when the patient goes. So you have to have at least 5 controls and blood tests. And for clinician, for us, it's not good, because then we might have weekend transfer on weekends. But if the patient is natural cycle, of course, she might. It seems like it's not very convenient, but when it comes to outcome, now this is a study from 2019 from Sweden. Now they analyzed close to 10,000 cycles of frozen embryo transfer, the natural cycle and artificial cycle. The artificial cycle is not good, because they have higher complication of of obstetric and pregnancy complications, which these four are the major ones. The hypertension of pregnancy, cesarean section is high, hemorrhage is high, and macrosomia. They tend to have larger babies. And this one, I'm going to show you another publication. This is the meta-analysis. This meta-analysis is like combined several randomized controlled trials. It just published a couple months ago in one of our journals, and it included 26 randomized controlled trials and 113 cohort studies. So it's like close to 150 studies, combination of 150 studies. And again, same finding, some of the obstetric complications were high in natural cycle, the hypertension of pregnancy, postpartum hemorrhage, and very preterm birth was high, maybe because of hypertension of pregnancy. But in natural cycle, in this study with combined data, the live birth rate was higher. So it's good. Natural cycle is good because there's less complication of pregnancy, and you are more successful. That's why I think even it might be, I mean, more disturbing because you are going to have, instead of just one visit, you will have 3 to 5 visits, and you cannot make a plan when the ovulation is going to happen, when my transfer is going to happen. But on the sake of your success and for your pregnancy health, I think natural cycle seems better. That's why I think this is my last slide. I think we have to go back to the nature. We should do freeze roll. We must have as much as eggs and as much as embryos. And I think when we do transfer, we should prefer natural cycle instead of artificial cycle. Thank you very much for listening to me and for your patience. And if you have questions, of course, we will later discuss. Thank you.
Thank you very much, Kemal, for a wonderful presentation. And we will take the questions after about 15-20 minutes. Let me finish my talk and then we will answer the questions. Thank you very much.
Thank you.
I will be quick, another 15-20 minutes. And so today's webinar is for patients, and I will try to be as lucid as possible. And the topic chosen is the globally the most common intervention for fertility, or the most common fertility intervention. In 2018-19, there was a survey done only on the Indian subcontinent, and the survey said almost 18 million IUIs were done only on the Indian subcontinent, with 45,000 registered gynecologists and possibly about 15,000 unregistered gynecologists. So this is really a large number, and patients are very keen to know what is the value of IUI, what are the success rates. So I will try to break this down into a small, simple presentation. So what is IUI? IUI is the therapeutic introduction of processed sperms, capacitated sperms. Capacitation means making the sperm imminently fertilizable. That means certain chemical reactions happen in the head of the sperm, which makes the sperm immediately fertilize the egg as it meets the egg in the mid-ampullary portion of the fallopian tube. So what we do is we actually do whatever happens usually in the body, in the cervical canal. We do this capacitation outside in the laboratory and then introduce this capacitated sperms in the upper uterine cavity. Essentially, what we are doing is we are mimicking the function of the human cervix. So what is the function of the human cervix? Cervix is the mouth of the uterus. So the function of the human cervix is to produce cervical secretions. Because of these secretions, the sperm remains alive for up to 72 hours after ejaculation into the vagina. It acts like a physiological filter and removes the dead sperm, epithelial cells, the other, hair, all the other things, the shed things is all cleaned by the cervix. So only the sperms go up into the cervical crypts, In the cervical crypts, these sperms are stored for 72 hours and are thrown up in asynchronous waves over the next 72 hours towards the internal tubal ostium in the upper part of the uterine cavity. So this is the actual function of the human cervix. So what do? We carry out, we mimic the function of the cervical, of the cervix in the lab. So we get rid of debris, abnormal sperm, seminal plasma. Out of the good, bad, and ugly sperms, we pick up only the good motile sperms. And in vitro means outside the body. And in the laboratory, we using some culture medium, some chemicals, we do an in vitro capacitation of the sperms, making them imminently fertilizable. So what are the indications of IUI? So these are the traditional indications: unexplained infertility, cervical infertility, male infertility, retrograde ejaculation, sometimes after transurethral resection of prostate, the male lands up with retrograde ejaculation, and then we give a alkaline fluid to the male. He drinks it. When he ejaculates, the sperms, they land up in the bladder and the sperms remain alive. After ejaculation, we ask the male to pass urine in a jar and then recover the sperms from his urine. It's got very high success rates. Immunological infertility, if there are anti-sperm antibodies, mild endometriosis, very rarely allergy to seminal plasma, multiple failed IVF. Sometimes IVFs keep failing and we go back to doing IUIs. And simply in the developing world and the economically poor countries, inability to afford IVF. Contraindications to IVF are unhealthy tubes, acute genital infection, hyperstimulated ovaries are relative contraindications in countries like UAE, where I practice. If you have more than 3 follicles, you have to cancel the IUI cycle and ask the couple to plan intercourse at home. Medical, psychological, social indications, contraindications. Sample collection is by masturbation. A split ejaculate is preferred, where the first part of the ejaculate is collected in one jar. The first spurt and the second, third, fourth spurts are collected into another jar. Basically, 90% of the sperms are in the first part of the ejaculate, and we send the partner or wife in to help the husband collect the ejaculate completely. Viscous sample can be treated in the laboratory very easily. And for those who have very low counts and they cannot afford ICSI, and we pool ejaculates, we ask the man to ejaculate 2 or 3 times over 6 to 8 hours, pool the ejaculate and try to collect the sperms and then do an IUI. So essentially the sperms after collection, I've told you we do some sort of capacitation outside. So the capacitation is by swim up methods or density gradient separation. And very recently, we have come up with microfluidic sperm sorting chips, which do the sperm separation and capacitation. And the picture on your slide, the A and B and C and D are the microfluidic sperm sorting chips. And this is in our lab, the sperm sorting chip being loaded, preloaded with culture media and then loaded with sperms. So you put the sperms at one end of the chip and the motile sperms can be collected from the other end. Today, the microfluid sperm sorting method significantly increases the ongoing pregnancy rates as compared with the older methods that we used to do. And this is a study to predict the value of post-wash total progressively motile sperm count. And so if the total progressively motile sperm count is between 0 to 0.5, 0.5 to 0.1, 1 to 5, 5 to 10, and greater than 10, IUI works best when the total progressively motile sperm count is at least 5 million. So if it is more than 5 million, then that is the least that is required, total progressively motile sperm count, not the total sperm count. This is a slide that actually tells you how post-wash a sperm sample, a capacitated, clean, forward progressive sperm sample that is used for IUI looks like under the microscope. And when should IUI be done? IUI if the patient can afford it, then we do 2 IUIs, 24 to 40 hours. The logic behind it is before and after the egg release. Remember, the sperm meets the egg in the mid-ampullary portion of the fallopian tube, and the timing of the intrauterine insemination is done accordingly. And if you have to do just 1 IUI, we prefer to do it 40 hours after the HCG trigger. Today, there are a lot of trials that tell us that it's not 36 hours, but 40 hours after the HCG trigger gets us the highest success rates. For all the patients, we have almost 104 patients attending this webinar. As I'm speaking, you can put your questions not in the chat box, but in the question and answer box. I don't have access to the chat box and I will be unable to answer any questions in the chat box. Please start writing your questions for both the speakers only in the question and answer box. And this is a study from Israel that tells us, does optimal follicular size in IUI cycles vary between tablets, clomiphene citrate, and injections, fertility injections? Yes, they vary, and in 16 to 18 millimeters is the trigger with fertility injections. And when you use clomiphene, at least when the leading follicle reaches 22 millimeter, one should trigger. Also, please do not cancel cycles if you don't get that 8 millimeter. It's a wrong notion that endometrial thickness has to be 8 millimeter. Endometrial thickness cannot predict ongoing pregnancy rate in IUI cycles stimulated with clomiphene citrate. Also, this is an old paper, but I still believe it's true. 2 inseminations before and after ovulation, it gives us a higher pregnancy rate. But remember, an IUI pregnancy rate is never more than 25% when you compare it to IVF. The success rates are 65 to 70%. This is a very old paper from Canada, from University of British Columbia, that tells us that you must go through IUI a couple of times and then move on to IVF. So what are the reasons for these high pregnancy rates when you use fertility injections with IUI? So the increase in ovarian size brings the ovary closer to the fembryal end of the fallopian tubes. And the swim-up techniques enhance the fertilizing capacity. IUI increases the number of sperms reaching the mid-ampullary portion and increases the chances of fertilization. When is IUI not beneficial? IUI is not beneficial to women over 40 and has the best chance of success within 3 cycles. Also, using fertility injections is better than using tablets when you are attempting IUI. Again, this paper systematic review tells us using fertility injections and IUI is better than tablets and IUI. Again, there is no advantage of using letrozole. I prefer clomiphene when the only option is to use tablets because of economic reasons. Otherwise, for highest success rates, one should use fertility injections. Do not repeat IUI more than 2-3 times if done using fertility injections. Again, no more than 15 minutes bed rest is required, and do not process the sperm sample and keep it on the shelf or in the incubator for more than 60 minutes. Once the sample is processed, please ask your doctor to, even if he's very busy, to put the sample in, do an IUI within 60 minutes of processing. Like any other medical procedure, IUI has complications. You can, if done not in a clean environment, if the sample is not processed under a laminar flow, you can have infections, and these infections can be terrible. I have seen in early days peritonitis following an IUI when things were not retrieved. On the other hand, this is a paper from the USA, from Chicago, from '94, where the man had a ureterorectal fistula. Every time he was ejaculating, the sperms were going into the rectum, and the team was with a glass pipette getting the sperms out of his rectum, washing them thrice with antibiotic supplemented medium, and he conceived 2 children using sperm with IUI, with sperm retrieved from the rectum. So what are the take-home messages for success? Do not inseminate over 0.5 ml processed sperm. The softer the catheter, better the results. The IUI procedure must be finished within 2 minutes maximum time. One must do ideally under a partially full bladder with transabdominal guidance, sonography guidance. And if there is blood inside or outside the IUI catheter, or you find it on your panties later, the chances of pregnancy go down. Thank you for a very patient hearing.